Welcome back. Today we're doing a deep dive into a bacterium that, let's be honest, many of us in the lab have historically pushed to the side, Corynebacterium mucifaciens. For a long time it was just seen as a simple skin bug, a contaminant, but that view has completely changed and it's now an emerging pathogen that absolutely demands our attention.
This quote right here, it perfectly captures the massive shift that's happening in clinical microbiology. For decades, we lumped organisms like this into the diphtheroid bucket and pretty much ignored them. But that way of thinking is completely obsolete now. And it's really down to two big things. We have way better technology to actually identify them, and our patient population is changing, with more immunocompromised people and more indwelling medical devices. We just can't afford to ignore these bacteria anymore.
So to put this whole story together for you, we synthesized a ton of research using Gemini Deep Search. And if you're the kind of person who wants to get your hands on the primary literature, don't worry, the full list of sources is right there for you in the notebook document that comes with this explainer.
Okay, so here's our game plan. First, we'll talk about that big paradigm shift in how we see this organism. Then we'll walk through the modern lab workflow for IDing it. After that, we'll look at the actual diseases it causes, dive into the really tricky treatment landscape, and wrap up with infection prevention.
All right, let's kick things off with this fundamental change in perception. This is really a story about an organism's journey from being tossed in the diphtheroid bucket to being recognized as a specific, clinically important species.
You know, this slide really tells the entire story in a nutshell. Over on the left, you've got the old view. Just another diphtheroid, probably a skin contaminant, no big deal. And honestly, that was mostly because our lab methods just weren't good enough. But now, look at the right side. That's the new reality. With amazing tools like MALDI-TOF mass spec and gene sequencing, we could nail the ideas Corynebacterium mucifaciens and link it directly to some really severe invasive diseases. It's a perfect example of how better tech uncovers threats that were hiding in plain sight.
So what is this bug, really? Well, it's a gram-positive rod. And under the microscope, it has that classic choreoniform or pleomorphic look, which just means the cells can be all sorts of irregular shapes and sizes. And its name, Mucifaciens, it literally means slime-producing. And that's a huge clue, because it often forms these classic mucoid, slimy-looking colonies on an agar plate. That's a key visual hint for us at the bench. Okay, let's move over to the lab bench and see how this all comes together. Because like we've said, getting an accurate ID isn't always a walk in the park. This is where the modern microlab really shows its worth. Now this is a hugely critical point for every single person working in a clinical lab. We might be looking for those classic, slimy, yellow colonies, but we absolutely cannot depend on it. Atypical, non-mucoid strains, the ones that just look like any other corneoform, have been pulled from fatal bloodstream infections. This proves that just looking at the colonies isn't just not enough, it can be dangerously misleading.
So this reality means we need a structured workflow. Step 1 is your presumptive ID, based on the gram stain and what the colonies look like. Step 2 is confirmation with MALDI-TOF, and the key here is that you must do a protein extraction. Direct smears are just not reliable for this group. Then finally, for any critical isolate, we're talking blood, CSF, a prosthetic joint, you have to reflex to the gold standard, sequencing a housekeeping gene like RPOB to get an absolutely definitive identification.
Okay, so let's connect the dots from the lab to the patient. We can identify it, great, but what diseases does Corynebacterium mucifaciens actually cause? Well, the list is probably a lot broader than you think.
So bloodstream infections and sepsis are what we see most often, especially in patients with central lines or other catheters. But it definitely doesn't stop there. It can cause some nasty respiratory infections, even necrotizing pneumonia. And once it gets into the blood, it can travel, causing metastatic infections like septic emboli in the brain that can actually look like tumors on a scan. We also see it pop up in localized infections, everything from wounds to prosthetic joints.
This case right here is a total game changer. Why? Because it just shatters the idea that only the severely immunocompromised are at risk. This patient was immunocompetent, but they had heavy exposure to a farm environment, which we know is a reservoir for this bacterium. This tells us that if the inoculum is big enough, it might be able to overwhelm even a healthy immune system, and that makes Corynebacterium mucifaciens relevant to a much wider group of patients.
All right, so we've got an ID and we've got a clinical syndrome. The next big question is, how do we treat it? And this is where things get incredibly complicated because we're working in what you could call a major guideline vacuum.
This data from a big surveillance study really makes the challenge crystal clear. Up at the top, you see vancomycin and linezolid, both at 100% susceptible. That makes them your reliable go-to drugs for empiric therapy, when you're starting treatment before you have the lab results back. But then, look at the bottom half of the table. For common drugs like penicillin and Cipro, resistance is rampant. The message here is loud and clear. You cannot guess. You absolutely have to do formal antimicrobial susceptibility testing.
And for those of our colleagues in labs that follow UCAS guidelines, there's another layer of complexity. There are no species-specific breakpoints for Corynebacterium mucifaciens. What that means is that labs have to interpret the MIC values using the general breakpoints for the entire genus Corynebacterium species. This makes the expertise of the clinical microbiologist, the person who can put the results into context, using expert rules and epidemiological data, absolutely essential for guiding treatment.
So, how are clinicians supposed to move forward? Well, we can look at two different approaches here. On the left, you have the Dutch swab guidelines. Now, they're not specific for this bug, but they offer a great framework from their prosthetic joint infection guideline. Start with vancomycin, then de-escalate to high-dose benzophenylin if it's susceptible, and they even give options for an oral step-down. On the right, the broader international consensus is more about principles. Start with vanc or linezolid, but after that, treatment is strictly dictated by the lab's AST report, with a huge emphasis on source control, like getting that infected catheter out.
Now, what about pediatric patients? The situation there is even tougher. There are zero formal guidelines. Treatment is completely extrapolated from adult data and a few case reports. This means individualized, lab-driven therapy isn't just a good idea, it's an absolute must. The principles are the same, sure. Empiric coverage, then targeted therapy. But every single decision, especially dosing, has to be carefully tailored to that child-specific age and weight.
And finally, with a pathogen that literally lives on our skin and in the environment, how do we stop these infections from happening in the first place? Well, the focus is very, very different from its more notorious cousin, Corynebacterium diphtheria.
The absolute key thing to understand here is that prevention is all about stopping auto-inoculation. It's about preventing the patient's own bacteria from causing an infection, not about stopping it from spreading person to person. So, that means standard precautions are king, especially meticulous hand hygiene. And strict aseptic technique when you're placing or maintaining any kind of invasive device is completely non-negotiable. But unlike diphtheria, you don't need to put these patients in droplet or contact isolation. Standard precautions are all you need.
And this really is the main takeaway from all of this. In this guideline vacuum for Corynebacterium mucifaciens, the role of the clinical microbiology lab becomes more important than ever. That antimicrobial susceptibility report is not just a piece of paper with data on it, it becomes the primary directive. It's the de facto guideline that allows clinicians to prescribe rational, effective, and targeted therapy for their patient.
We know that we have an incredibly knowledgeable audience, full of microbiologists, lab technologists, and infectious disease docs. Your expertise is invaluable, and we want you to help our community grow. So please, subscribe to the channel, and more importantly, share your expert feedback and your own experiences with this organism down in the comments.
So this all leaves us with one final and, I think, pretty provocative thought. The whole story of Corynebacterium mucifaciens is a story of reevaluation. It really makes you wonder, which organism that's sitting in our labs right now, being dismissed as just another contaminant, is going to be the subject of our next deep dive. Thanks for watching.